These are all the people that have helped me in my journey to standing here before you today. Thank you very much indeed for your generous introduction. As you've said, it's an extraordinary honour for me to be delivering the Sharpey Schafer lecture as an endocrinologist, something I couldn't have dreamed about when I was beginning my research work. Today, I'm going to talk to you about two key questions in human reproductive biology. And in fact, my work on this started more than 25 years ago. It started 36 years ago as my wife, Annie, who's here, was delivering our twins. In between rubbing her back and trying to be a supportive husband, I thought, why has Annie gone into labour now? And why has this pregnancy lasted 9 months? What determines these events? And I knew that at the time I was asking those questions, nobody knew the answer to these questions. And yet, channeling Kevin Tong after his brilliant presentation on Monday, humans had already landed on the moon at that stage, and yet we didn't know how we ourselves give birth. What a challenge for physiology. And this isn't just an academic question. It's a life-threatening question. If you're born too early, your chances of dying go up dramatically. If you're born too late, your chances of dying go up dramatically. So getting the timing of birth right is critical. And it's not just about birth events either. It impacts on the trajectory for life of the child. And this is data from Scotland showing that the longer a gestation goes on for, the less likely a child is to require special education facilities. But to try and answer our first question about what determines the length of pregnancy, we actually have to go back a long way, a lot longer than 36 years, more like 320 million years, to the evolution of the amniotes. Prior to amniote evolution, we have amphibians, and as you'll see here, and I'm sure you all know, amphibians fertilize their eggs outside the reproductive tract. This means that the reproductive tract is in constant contractile activity. But with the development of the amniotes and the formation of eggs, this changes. With the dinosaurs, we have internal fertilization. And then we have the formation of an egg and sometimes even viviparity. The key change for me that's occurring in this process is that the reproductive tract has to have its previously continuous contractile activity interrupted to allow the development of the egg. Something has to put the brakes on that contractility. You say, how does Roger know anything about pregnancy in dinosaurs? It's not easy, but we do have some fossil evidence. Shown here is a pregnant ichthyosaur. And remarkably, It was fossilized in the process of delivering its live young. It's likely that this process of reproduction in dinosaurs is very similar to birds, and progesterone plays a key role in stopping the contractile activity of the reproductive tract to allow the formation of the egg. Humans too, of course, have internal fertilization, and then the development of the embryo after implantation within the uterus. This too requires the reproductive tract to stop its contractions. The second problem, however, with the evolution of mammalia is how do you tolerate the fetal allograft? The answer is with a virus. Endogenous retroviruses form about 6% of our genomes. Past viral infections of retroviruses into germ cells and incorporation within our DNA in germ cells has allowed these retroviruses to be perpetuated every time we reproduce. And Richard Dawkins might say, we are, in fact, no more than a complex way for a retrovirus to reproduce. And you could argue again for Kevin Fong that we have allowed retroviruses to go to the moon, and soon retroviruses will go to Mars. Retroviruses in our germline were inserted at the very beginning of the formation of placental mammals. Every placental mammal that has been studied has retroviruses, and these retroviruses particularly get expressed within the placentas. Their expression is allowed by the changes in DNA methylation that occur following fertilization in the zygote. Particular retroviruses have inserted in the past history of primates. Syncytin 2 is found in all the monkeys, both the Old World and the New World, but not in lemurs. That insertion probably happened around about 25 to 30 million years ago, and more recently, Syncytin 1 has infected our ancestors about 10 to 15 million years ago. It plays a critical role within the blastocyst, driving production of placental chorionic gonadotrophin, which in turn drives progesterone production from the ovary. And progesterone and HCG together combine to stop the contractions of the uterus that allow implantation to occur in humans. This Syncytin 1 is shown here in pink within the human blastocyst. Here you can see the beautiful pictures created in this publication with the pink area representing the implantation site with high levels of the retrovirus coat protein Syncytin 1. The lemur has a hemochorial placenta. It lacks Syncytin 1 and Syncytin 2, a very different structure to our own placentas, where this layer of syncytiotrophoblast formation caused by fusion of cytotrophoblasts to the syncytiotrophoblast, mediated by syncytin 1 and syncytin 2, allows this multinucleate layer to be produced, where HCG is formed. HCG can actually form a positive feedback circuit with its own production via cyclic AMP. And linked into this cyclic AMP stimulates corticotropin-releasing hormone production, or CRH, as shown on this slide. These positive feed-forward circuits are unusual in human biology because they lead to exponential increases. And so we see across pregnancy an exponential increase in the number of syncytiotrophoblast nuclei. Paralleling this increase, we see an exponential rise in placentally derived corticotropin-releasing hormone. In this slide, you'll see the rapid rise that occurs in patients who will subsequently deliver prematurely, and the slow rise that will happen for individuals who deliver post-8s. This is a kind of biological clock located within the placenta that regulates the length of human gestation. So our first question about what determines the length of a human pregnancy is linked to a maturational process occurring within the placenta in which corticotropin-releasing hormone and cyclic AMP and a retrovirus plays a critical role. Our lives are determined by a retrovirus. What about the onset of labour itself? How does that work? As Mike Taggart mentioned, we can look at different species and gain clues. But in reproduction, it's particularly difficult because each species reproduces differently. On this slide, you can see examples of a number of different species of mammal, where the onset of labour is precipitated by a fall in estrogen, a fall in progesterone, or a rise in estrogen. But look at the curves for the human. Progesterone and estrogen rising across pregnancy and not falling at all until the delivery of the baby. What's happening in the human? Primates are also unusual in having multiple estrogens. Humans actually have 4 during pregnancy, but 2 are dominant, estriol and estradiol. Why have we got all these different estrogens? Where are they coming from? Estriol actually gets made in the placenta from precursors from the fetus. DHEA from the fetal adrenal is 16-hydroxylated in the fetal liver before being converted into estriol. Estradiol, on the other hand, comes predominantly from maternal adrenal production of DHEA and placental formation of the estradiol. Late in pregnancy, CRH and estriol levels correlate closely. The reason is that CRH made in the placenta is released not only into the mother's blood, but also into the fetal blood. And CRH, we've shown, can directly stimulate DHEA production from the fetal adrenal. But let's look at the estrogen and progesterone again in human pregnancy. Here are the curves showing the changes across gestation. What do you make of it? Where's the signal? I could see no signal. And then a paper came out from Melamed showing that estriol and estradiol are actually mutual antagonists at the estrogen receptor, the ER alpha. So here you can see the interactions between estradiol forming homodimers, interactions with polymerase, and the formation of estrogen target genes. Equally, estriol can interact with ER alpha, form homodimers, and stimulate transcription. But heterodimers do not initiate transcription. So once you have that information on board, the pattern that you're interested in changes and ceases to be the direct increase in estriol and estradiol, but more the ratio of these different steroids. And then an insightful pattern emerges where the ratio of estriol to progesterone falls across pregnancy. Progesterone to estriol falls across pregnancy. So progesterone is becoming less dominant. Equally, the ratio of estriol to estradiol increases progressively across pregnancy. And these patterns continue right up to the time of delivery. So we now see that the placenta, through production of CRH released into the fetus, is driving this increase in estriol, which in turn is driving the processes that lead to the onset of labour. The effects become even more dramatic because right at the end of pregnancy, CRH binding protein, which modifies its effect, falls. So the biological effect of CRH increases, explaining this correlation that we've already looked at between estriol and CRH in late gestation. Of course, when you find a result like this, you want to pursue it with an animal model. So we studied baboons. We were puzzled because our beloved CRH hormone peaked in the middle of gestation. How could it be doing that and causing the onset of labour? We thought perhaps there was something strange about the baboon. So we studied the New World monkey, the marmoset, and again, levels peaked in the middle of gestation. What was happening? What's going on? But then I looked into the estrogen levels within these monkeys and discovered that indeed there's a pattern in the monkey with estradiol peaking at the same time as the CRH and then falling, while estrone levels rise late in gestation. And if you think again about the importance of ratios of these different estrogens, it starts to make some sense. We need a dominant estrogen to arise within the circulation and affect the myometrium. In humans, it's estriol. In monkeys, it's estrone. And this mixing of the two generates a signal. But it turns out the picture is even more complicated. The ER alpha gene, that is the dominant gene within the myometrium for estrogen receptors, is a complex gene. And you can form different isomers by different splicing events. ER alpha can interact with estradiol, or estrone, or estriol to cause transcription in the way that we've discussed. But there's also a delta 7 isoform. And the delta 7 isoform is present in the myometrium for most of pregnancy. And heterodimers are not active in transcription. So this is a way that estrogen action in the myometrium can be blocked until the time of labour. And crucially, humans always go into labour. Now you might think that's not unusual, but it is. Many other mammals have defects, for instance in the sheep, that prevent labour. If a fetal sheep doesn't have its adrenal gland or its hypothalamus, it doesn't go into labour at all. And the sheep ewe eventually dies with a giant fetus. Doesn't happen in humans. So we've seen that different ratios of estrogen can cause the onset of labour. Both in New World monkeys and Old World monkeys, changes in the ratios cause the onset of labour. Why? These higher primates, monkeys and humans, the apes, have long-lived mothers with huge investment in bringing them to sexual maturity. This is a mechanism that allows the mother to be preserved even if the fetus dies. It's normal for labour to occur due to high levels in humans of estriol. But if the fetus dies, estriol levels plummet and estradiol will become dominant and cause the onset of labour. And a similar sort of change occurs in the monkeys. But what about progesterone action? Progesterone maintains pregnancy. How does it do that? Well, it does it by interacting with the progesterone receptor. And again, there are alternative start sites for the progesterone receptor, leading to alternate products, PRB and PRA. When progesterone interacts with PRB, which is the dominant force for most of pregnancy, you get the formation of dimers, and you get the formation of an inhibitory complex on the DNA, preventing transcription of key genes, such as the gene for connexin 43. By inhibiting connexin 43 expression, you reduce the likelihood of contractility within the uterus. But late in pregnancy, probably partly under the action of increased estrogen, PRA levels increase. And also there's the formation of 20-alpha hydroxysteroid dehydrogenase. Levels of this enzyme increase in the myometrium and catabolize the progesterone. The unliganded PRB is inactive. The unliganded PRB is inactive. But unliganded PRA is in fact effective at stimulating the transcription of connexin 43, allowing increased contractility. So putting all of this together, we have, at the beginning of pregnancy, increased concentrations of HCG, driven, as we've seen, by syncytin. These increase progesterone production and cause inhibition of contractility. But they also lead to increased levels of CRH and cortisol, which forms a positive feedback circuit. As the levels of CRH and cortisol rise, maternal production of ACTH goes up, maternal cortisol, and likely DHEA. There's also the stimulation of the fetal production of DHEA. These interact to form estrogen, but they're blocked by the action of progesterone receptor B, which inhibits the expression of estrogen receptor alpha. As CRH increases further, we get more and more estriol formed. And high levels of CRH then induce the production of NF-kappa B and prostaglandin F2 alpha. These stimulate PRA production and inhibit progesterone. Now that PRA levels goes up, we inhibit the function of PRB, and with that, we withdraw the relaxation effect, the braking effect on the uterus. Now, ER alpha can act to stimulate connexin 43, the formation of COX-2, which increases contractility, and the inhibition of potassium channels, which also increase contractility. Then we have the onset of labour. This is an emergent property, best exemplified by the behaviour of a crowd at a soccer match. We've been working on how the uterus develops regular, organised contractions, a biological problem in which energy plays a key role. Disorder, such as you see on my desk, is what you expect in this world because of the second law of thermodynamics, which says that entropy or disorder will gradually increase. And if we look at the surface of Mars, we can see what this means with total disorder. And yet, when we look at the surface of Earth and coming closely, we see quite the opposite. We see high levels of order. How can this order be generated against the second law of thermodynamics? The answer is life. Life captures the energy from the sun and creates order. How exactly life began is unclear, but one possibility is that the alternating light and darkness created by the Earth's rotation creates periods of cold and warmth. This could be seen as being like a giant PCR machine that can melt RNA and then allow it to freeze again with a doubling of the amount of RNA with each rotation of the Earth. Over millions of years, this may have created the necessary substrate of RNA to allow the development of life. However it began, life uses the energy of the sun to replicate proteins, RNA, and DNA. And this allows the capturing of the sun's energy and the use of that energy to create the order that we see around us on our planet. We use energy to create the order seen in our bodies. The highest levels of order occur in our brains and our hearts. And during pregnancy, blood flow increases dramatically to the uterus to allow the generation of order within the fetus. Between the 10th and the 26th week of pregnancy, 250,000 neurons are added to the baby's brain every minute. This is an incredible demonstration of order and must have energy to create that order. The body also invests heavily in order within the heart, and within the heart, a key aspect is regular, sustained contractions that pass blood through the heart, carrying energy to the rest of the body. As I speak to you, my heart is contracting regularly in a very structured, organized process, as shown by my cardiac ultrasound. This order requires a pacemaker, which then directs the contractions of the atria and the ventricles. A highly ordered process. But let's think about the uterus. The uterus
also has to generate regular, ordered contractions. And yet the uterus has no pacemaker. How can it generate organized, regular contractions without a pacemaker? For the majority of pregnancy, the uterus is relaxed. It has a high level of symmetry. There are no big changes going on. And yet, as we get into labor, it transforms into an organ that has regular, sustained, synchronous contractions, which develop enough pressure within the uterus to expel the baby through the narrow pelvis and into our world. We think the way this happens is related to what are called coupled oscillators. Coupled oscillators are units that can be engaged into synchronous behavior, a bit like pendulums on grandfather clocks. Each muscle cell of the uterus can be thought of as a potentially coupled oscillator. Early in pregnancy, the muscle cells are quite separate from one another, and they don't exhibit coupled oscillatory behavior. But late in pregnancy, under the increasing influence of estrogen and with the withdrawal of progesterone action, this all changes. The cells become linked to one another by what are called gap junctions made of the protein connexin 43. Also, the resting plasma membrane potential changes so that it becomes easier for the cells to depolarize and fire off. In addition, changes in potassium channels means that depolarizations can last longer. These factors together allow the muscle cells to start to become linked together so that when one cell fires, the one next to it is more likely to fire. As pregnancy gets closer to labor, the number of gap junctions increases quite dramatically, allowing more and more muscle cells to be linked together into areas or patches within the uterus. When these patches get big enough, when they contract together, it causes an increase in the intrauterine pressure. An increase in intrauterine pressure will also cause stretch on the remaining muscle cells. And stretch is a powerful stimulus to contraction by muscle cells. This can allow all of the muscle cells within the uterine wall to become linked together into synchronous behavior to increase the intrauterine pressure. That in turn causes the delivery of the baby. A way to think of this is like the behavior of a crowd at a soccer match, where without any conductor, the crowd can begin to sing together, and eventually the whole of the crowd at the soccer match can sing a song together completely without a conductor. They've engaged like coupled oscillators, each member of the crowd listening to the people around them to gain cues on when to sing. The loudness of an individual singing allows people nearby to hear in the same way as the strength of a depolarization, and the connectivity of the muscle cells allows muscle cells to come together for synchronous behavior. This is quite different to the behavior of the heart, which is much more like the behavior of a conductor with an orchestra, where the conductor gives signals to the orchestra to allow them to behave in a synchronous and ordered manner. He can organize changes in the tempo, he can organize changes in different parts of the orchestra, but that is very different to the uterus, where coupled oscillatory behavior can allow the emergence of robust, powerful contractions without any pacemaker. Evidence for this view of uterine action comes from the work in Little Rock, Arkansas, where the SARA facility allows superconducting magnets to identify depolarizations within small areas of the uterus. Their data shows this development of synchronous behavior across patches of the uterus, eventually spreading to the whole of the uterine wall as the woman gets closer to delivery of the baby. This is a video to go together with our paper, Why the Heart is Like an Orchestra and the Uterus is Like a Soccer Crowd. And I'm speaking to you on behalf of my co-authors, David Banney, Roger Young, Mohammed Imtiaz, and Jonathan Paul.
So this coupled oscillatory behavior develops. But how do we study the molecular basis of these events when we can't experiment on pregnant women? And yet animal models do not replicate the human situation. Each woman can only possibly give us one sample. How can we put those samples together to try and understand the process? We've done that by taking samples from pregnant women in labour and not in labour and performing what's called subtractive hybridization. We've identified 30 genes that increase in samples associated with labour. And we've done then quantitative PCR for these 30 genes in 60 subjects, 30 in labour and 30 not in labour. This shows our results. And once again, it's a challenge to interpretation. But as you look at the results, it becomes clear that in some individuals who are in labour, they'll be expressing large amounts of certain genes, and not at all the genes found not in labour. But for other genes, they may be expressed before the onset of labour, even though they're a characteristic of labour. So you can gradually develop a pattern across these 60 subjects. Using radar plots, for instance. And you can see on the left, a woman not in labour. In fact, number one, the one that had least expression of all the labour-associated genes. On the right, number 60, who had most expression. But you'll also notice that no individual gene stands out. You can put this together into what we've called a cigar plot of the 60 genes. And you can see how clearly the labouring group differentiate from the non-labouring group, and yet it's not based on an individual gene. More, it looks like a shotgun with increased gene activation as labour progresses. And this is the turning on of many genes that are in fact consequent of labour. Genes for metabolism, genes for inflammation, genes for repair. But the actual onset of labour appears to be an emergent event generated by many genes downstream from estrogen and progesterone signalling. We've been starting to try and translate this information into improved therapy. And one of the ways we've done that is to create nanoparticles targeted to the uterus. We've done that using nanoliposomes, which we've coated with antibodies to the oxytocin receptor. These nanoliposomes can then carry drugs directly to the uterus.
So Roger, where are we heading?
Jonathan, we're heading off to the uterus today.
Excellent.
So the challenge for us is to get the uterus specifically, a bit like organising a GPS on a car. So how do we do that, Jonathan?
We're doing that through using targeted nanoliposomes. In this case, the targeting is achieved through conjugating the oxytocin receptor antibody to the surface of the nanoliposomes.
That's cool. So the advantage of doing this is that we can deliver the drug just to the muscle cells of the uterus, and that has some advantages, doesn't it?
It certainly does. It has the advantages of improving the efficacy of the drug while simultaneously reducing the likelihood of off-target side effects.
So we've seen that the particles don't cross over to affect the fetus or the mother's brain.
That's true. We have seen that the particles do not localize to these organs. However, the targeted nanoliposomes do localize to the uterine tissue of pregnant mice.
That's really helpful in potentially enabling us to use drugs like indomethacin, which might have an adverse effect on the fetus.
And we have already demonstrated that by loading our targeted nanoliposomes with indomethacin, we can actually prevent preterm birth in an LPS mouse model.
The future clinical applications of this technology include treating women with preterm labour, but also potentially using it to induce labour more safely and treat women with postpartum hemorrhage using different agents to increase contractility.
And on that note, let's go to the uterus.
This system required the manufacture of the nanoparticles by Susan Hua, the animal research conducted by Jonathan Paul, and the conceptual targeting which I suggested.
How far to the uterus now, Roger?
Oh, we're getting pretty close.
So I was going to finish with this slide, which again shows all of the people that have been involved in the production and filming of this work. But again, I was so excited by Kevin Tong's presentation on Monday at lunchtime that I thought that once again I should channel him. Thank you.